So up first, we are joined by Neuroscientific Biopharmaceuticals with the ticker code NSB for an update about their lead drug candidate, Empton B. To tell us more, I'm joined by Neuroscientific's Managing Director and CEO, Matthew Liddelow. Good morning, Matthew. How are you?
Hi, Jane. How are you?
Great. Let me bring up your presentation.
Great. Thank you very much, Jane. And thank you. Appreciate the opportunity to present today. And thank you all those who have come online to watch these presentations this morning. Please note the disclaimer that just briefly flashed up before, but we'll move into a brief overview of who we are and what we do at Neuroscientific. Now, we're a drug development company that's focusing on developing peptide-based therapeutics that have disease-modifying potential for neurodegenerative conditions. And neurodegenerative conditions are essentially diseases that damage nervous tissue, mainly of the brain. We have a lead drug candidate called Empton B, which Jane mentioned at the top of this webcast, which is a first-in-class treatment that we're developing for Alzheimer's disease, multiple sclerosis, and glaucoma. First-in-class does mean that it's the first of its kind to make it to market, if it does make it to market later and down the track. And more probably for us, the most exciting point for this company at the moment is the fact that we are transitioning Empton B into its first phase 1 clinical trial in the first half of 2022. We're looking a bit more from a corporate overview of the company. We listed on the ASX midway through 2018 with a market cap of approximately 15 million. Current market cap sits at approximately 42 million, and we have a net cash position of 9.5 million in the bank. And our board and management team is a good mix of technical expertise and also capital markets experience. Most notably, we had Paul Rennie, who is more commonly associated with Paradigm, but a very successful biotech executive. It joined the company as chairman midway through last year. We've also expanded our team to actually include some additional operational staff as we head into clinical development this year. A little bit more about the patient groups that we focus on. The biggest problem with neurodegenerative conditions is the fact that most of these diseases don't have a lot of treatment options available to them. And if they do, a lot of the drugs are not very effective. And a good example of that is Alzheimer's disease, in that currently you've got a selection of drugs that really only treat symptoms, and the bandwidth of research that's focused on is very, very narrow. Therefore, there hasn't been a lot of other options that have come onto market in the last 25 to 30 years. Multiple sclerosis is a little bit different in that you do have effective treatments for one part of the disease, which is the earlier part of the disease, but as that disease progresses and symptoms get worse, these drugs become less and less effective. So the biggest issue for addressing this problem really is the fact that you don't really fully understand what the problem is. And it's hard to develop a solution when you don't know what the problem is, but we're pretty confident in Neuroscientific that we've got a very good workaround to finding a solution to these problems. Our lead drug candidate, Empton B, is modeled on a human protective protein. So our research really does involve really developing mimics that essentially copy or stimulate the body's own defense mechanisms to fight these diseases or protect the tissue damage that's being done. And Empton B is modeled on a subset of a particular part of a human protein, and it does stimulate that protective effect. And it targets a receptor that is highly expressed on the cells in the brain. So it works across multiple different types of neurodegenerative diseases, even though the underlying cause of those diseases is not completely identified. So having a look at Alzheimer's disease specifically, we've been able to validate that defensive mimicking activity of Empton B in a number of models of Alzheimer's disease. And essentially, we've, even in the gold standard animal model, where you can see here, this is the data from that particular study in which there was a 70% increase in actual memory improvement, which indicates that we are having a protective effect on the tissue of the brain and maintaining the structures of those cells. Multiple sclerosis is a really exciting indication for us in that we are getting, say, develop a really large body of evidence there based on data that's impacting multiple different pathways of multiple sclerosis. And to date, we've been able to also show an effect on the inflammatory response. So we are potentially an option that could treat all aspects of what so far is thought of the pathology of multiple sclerosis in the earlier stages, where there's a really strong inflammatory response. And in the later stages, you get this degenerative response of the brain cells. So EmtinB has the potential to work across all those pathways and provide a solution to patients at all treatment stages. And another focus for us is looking at degenerative conditions of the optic nerve. And the optic nerve is essentially a host of nerve cells that transmit what you see from your eye to be processed by your brain and the connection there. And there's a number of conditions that affect that optic nerve. The most common one or prominent one is glaucoma, where they think the pressure build-up in the eye actually causes the cells at the back of the eye to be affected and to actually die or be damaged, and therefore you get a reduction of vision. And what we've been able to show in a large animal model, which was a pig model, and that's very important due to the fact that the structure of the pig eye is very similar to the human eye. And we were able to actually maintain the structure of those cells that form the optic nerve and be able to maintain the integrity of the optic nerve and transmitting of that signal mechanisms, which at the moment, in that actual disease space, there's variable treatment options, but nothing that really slows down the progression of the damage to the optic nerve. So our current development pipeline involves streams of research in neurology and ophthalmology, and then we have individual indications within those streams of research, with Alzheimer's disease being one of the leading indications for neurology. And then our glaucoma research is the leading indication for ophthalmology, and we do some exploratory studies as well. But one thing to note with these particular streams of research is the safety data that we are generating in the preclinical stages to allow us to enter into the clinical development stages can be reused in other indications. So we don't have to repeat that safety data, that preclinical safety data over and over. We actually, due to the mode of action of Empton B being the same in all those diseases, we can repurpose that data across many different indications. To get our research done to the level and the quality that we demand, we use the best companies in the world. Some of those are local companies that we use in Australia, and a lot of those companies are international companies that we partnered with. We've also set about getting in preparation for our first clinical study later this year, or in the first half of this year. We've put companies in place such as Linear, and Agilex and Omeka, companies that will support our clinical development phase of the company. And also, we've just been cementing things, part of getting ready to actually execute on the clinical study. And while we have a host of milestones that we are due to report on throughout this year, really the big one for us will be this executing on this first in-human phase 1 study in the first half of 2022. It's the one milestone that all our investors have been waiting for. Interested parties that are looking to invest at a certain stage and at a certain catalyst in the company's history. And this is one of the big ones for us. And so there's a lot of excitement and enthusiasm around the company as we are really very, very close to making this a reality very, very soon. And we'll provide further updates in the near future, detailing more aspects around this clinical study. And on that note, that wraps up the presentation from me for today. Thanks, Jane.
Well, thank you for that, Matt. Great presentation. Looks like you've certainly got quite a fair bit of news flow coming up. We've got a couple of questions come through, so I'll jump into them. So, has the drug been proven safe to use, and when do you expect to have an indicative cost for it?
The drug's been proven safe to use in animals, but we're about to take it into its first human clinical study to demonstrate that initial safety data in humans. And as far as costs, we have our own model costs, but it's a bit too early to talk about that. As things further develop, the costs change as well. But one of the biggest aspects in developing a drug is obviously looking at the feasibility of that drug once it's a marketable product, and obviously that's something that we've paid particular attention to with EmtinB as well.
Thanks, Matt. Just another one. So, can you go into the MOA for EmtinB?
Mode of action is based on, well, basically binds to a receptor that is expressed on the outside of cells. Not to get too technical here, but it targets a receptor that is very common to many different cells that are expressed in nervous tissue. And it's on the outside of the cells, and it's called the LRP1 receptor. Now that receptor does a host of things once something binds to it, but we bind to a specific part of that receptor, and that induces survival pathways and regenerative pathways within the cells.
Wonderful. Well, thanks for that, Matt. That's all we've got time for today. I encourage webinar attendees to reach out by the contact details on the bottom of the ASX releases if they've got any other questions. But Matt, thanks for joining us.
Thanks, Jane.